Well, Manuel, I'd like to thank you for the very kind invitation to speak. And what I'd like to do in my 15 minutes this morning is talk a little bit about how H. pylori should be managed, and also I'll end with some recommendations on how to manage premalignant lesions within the stomach. So essentially, all persons who are colonized with H. pylori develop coexisting gastric inflammation, and this is the strongest known risk factor for gastric cancer. And as endoscopists, this is a lesion that we do not like to see when we do our procedures. This is a distal gastric adenocarcinoma within the antrum. Adenocarcinoma of the stomach is a very important problem worldwide. It accounts for over 700,000 deaths per year. Fortunately, for people that study this disease, there are very well-described histologic steps that lead to the most common form of gastric adenocarcinoma, and that's intestinal-type gastric cancer. So the questions that we need to ask are, number one, can we treat H. pylori infection and stop the progression of this disease? And number two, where along this cascade is antibacterial therapy effective? Well, many of the studies that earlier looked at this did not show a significant difference between people that received antibiotics and people that did not. This is a study in China, a randomized controlled trial, and even though there seemed to be a difference in the incidence of gastric cancer, this was not statistically significant. However, when the investigators did an analysis after the trial, they found that indeed there was a difference if they treated people who did not have intestinal metaplasia or gastric atrophy with antibiotics. Now, one problem with this trial is that it only lasted 7 years, and we know now that longer follow-up periods are needed to really determine the effectiveness of antibiotics against this disease. And these are data from Palaio, Korea in South America, in which they treated people with antibiotics, and H. pylori was successfully cured. And they found that 12 years later, there was an improvement in their premalignant lesions. So it takes up to 12 to 15 years before we're really going to see an effect on anti-H. pylori therapy. One caveat is that I want you to keep this in the back of your mind, that people that received antibiotics but did not have clearance of their H. pylori infection still seem to have a benefit compared to people that did not receive any antibiotics at all. So we were very excited as cancer biologists to see this trial published 2 years ago in the Journal of the National Cancer Institute. And this is the Shandong intervention trial. And in this study, they treated patients with antibiotics and followed them for 15 years. And they found that, yes, indeed, now we can treat people with antibiotics and significantly reduce their risk of developing gastric cancer. And when these results were combined with results from other randomized controlled trials, there was a significantly reduced risk or benefit of treating H. pylori in these individuals. The problem is that only 1 to 3% of individuals who were infected ever develop gastric cancer. So there are different determinants that seem to increase the propensity for an infected person to develop gastric cancer. One of these is strain variation, and many investigators have tried to group or cluster H. pylori strains using sequencing techniques. This particular study showed that strains tended to group in clusters that reflected the phylogeographic origin of their hosts. At a more granular level, certain proteins that are produced by carcinogenic strains of H. pylori, such as CAG-A, can be injected into host epithelial cells, and they'll disrupt many different pathways within these cells, all that are important in gastric cancer. Well, host genotypes are also important, and for me, it seems like that these are inextricably linked with the location of gastric colonization in the stomach. Most persons that are infected with H. pylori only have mild gastritis that can involve the whole stomach. People that are predisposed to duodenal ulcers have antral predominant gastritis and high acid levels, while people that are predisposed to develop gastric cancer have a corpus predominant gastritis and low acid. So if we're looking for a molecule that may be important in gastric cancer, it seems like one that has to do with gastric acidity, and inflammation would be a prime candidate to study. And IL-1 beta was the first candidate gene that was studied in this process. It's pro-inflammatory, and it's a very potent anti-acidic molecule. It's 600-fold more potent than PPIs. Many studies, including AMAD-LMR, have now shown that IL-1 beta polymorphisms that increase production of this molecule increase the risk for gastric cancer, but it's only present in persons that are infected with H. pylori. If you then combine high-expression IL-1 beta alleles with CAG genotypes, then you can further increase the risk that a person has for developing gastric cancer, up to 25-fold over baseline. One study that came out recently that used a different approach was a discovery-based study, and this was published in JAMA, and this was a GWAS study. And investigators used populations from the Netherlands and Germany to identify 2 different loci that were associated with H. pylori seropositivity. And one of these loci encoded TLR1. So we're now beginning to understand that SNPs, or polymorphisms within immune response genes, are important in the development of infection and possibly gastric cancer. Environmental cofactors is something that we're working on actively at Vanderbilt. Salt is a risk factor for gastric cancer. We've been able to show that if you grow H. pylori in increasing concentrations of salt, you get a marked increase in the expression of this oncoprotein CAG-A. And in our lab, we've been interested in another dietary element, and that's iron. And we've been able to show that iron deficiency increases the development of gastric dysplasia and gastric cancer, twice as high as H. pylori infection in iron normal animals. And the reason this happens is because if you take H. pylori and make it iron deplete, it starts to make many more of these CAG secretion pili that translocate CAG-A into the host cell. So I think as researchers, we're now forced to not just compartmentalize things into little convenient categories like microbial factors, host factors, or environmental factors, but we're learning that we have to understand how these specifically interact with each other to really garner a more precise risk of gastric cancer. So I want to turn to some exciting new areas that I think the field is going. Now, I showed you this trial earlier, and this showed that if you treat people or give them antibiotics, then they do have a reduced risk of developing gastric cancer. But buried deep within this paper was the fact that about half of the people that were treated with antibiotics still had H. pylori infection. So this suggested somehow that antibiotics may be manipulating other organisms than just H. pylori. And so this is an important issue now. So what is the role of the microbiome, for example, in gastric cancer? If you look at the microbiota in the stomach of somebody who's infected with H. pylori in green predominates. There's less diversity of the microbiome in the stomach of a person who's infected compared to somebody who is uninfected. In mice, you can show the same thing. If you take uninfected mice and then treat them with H. pylori, you see a marked change in their microbiota. There's a bloom or an increase in the firmicutes population, and there's a contraction of the Bacteroidetes. But the important question is, does this have anything to do with cancer? And in fact, at least in animals, it looks like it does. So Jim Fox did a study in which they took germ-free animals, shown in green, and then infected them with H. pylori alone. There was no other bacteria or microbiota in these animals. And they found that 80% of these animals did not develop any cancer at all. And the ones that did only developed low-grade cancer. In contrast, if mice had a conventional microbiota and you then infected them with H. pylori, shown in yellow, most of these animals developed high-grade neoplasia. So it seems that the gastric microbiota collaborates with H. pylori to make it more carcinogenic. So I think that we have to refine the way that we think about how H. pylori induces or interacts in this carcinogenic cascade. I think early on there's an intense interaction between H. pylori and its host. I think over time gastric atrophy develops, there's less H. pylori, and this allows other bacteria to colonize the stomach, which may be important in the later stages of gastric carcinogenesis. So, are there additional reasons that we should really think about who we're going to test and who we're going to treat for H. pylori? Well, a disease we're all familiar with is esophageal adenocarcinoma, which emanates from Barrett's esophagus. Many studies, this is just one of many, have shown that there is a reciprocal or inverse relationship between H. pylori or gastritis and the development of Barrett's esophagus. And as you might expect, there's also a protective effect of H. pylori infection, especially CAG-positive strains, on esophageal adenocarcinoma. And so many studies have tried to replicate this, and if you do a meta-analysis, you find that there is a significant protective effect of H. pylori infection on esophageal adenocarcinoma. This does not happen when you look at squamous cell carcinoma of the esophagus. And the effect on esophageal adenocarcinoma really is specific for CAG-positive strains because you don't see the effect with CAG-negative strains. What about other diseases? Well, there's very enticing data from Marty Blazer's group showing that there's an inverse relationship between H. pylori and asthma. And this happens at an early age and with CAG-positive strains. I don't have time to go into these other diseases, but there are other disorders that seem to be reciprocally related to H. pylori. So who should be tested and who should be treated? Well, this is my list in 2015. Persons who have ulcers or maltomas, if there's a family history of gastric cancer, if they're going to be on non-steroidals for a long time, non-ulcer dyspepsia, prior to long-term proton pump inhibitor use, or any premalignant lesions in the stomach, they should receive therapy for H. pylori. So, what is the current recommended management strategies for people who have endoscopy and are found to have premalignant lesions in the stomach? I'll say from the start that there are no good data to support what I'm about to tell you. But this is what we have. So there was a group that was formed 3 or 4 years ago which tried to develop evidence-based guidelines for the management of people who have premalignant lesions. And this was based on existing data, and these recommendations were then graded. So what about patients who have atrophic gastritis or intestinal metaplasia who do not have dysplasia? What should we do with those individuals? Well, after endoscopy, 4 biopsies should be taken, 2 from the antrum, 2 from the corpus. And from those biopsies, you can determine whether the patients have mild to moderate atrophic gastritis or intestinal metaplasia, or what we call extensive disease. If they only have these premalignant lesions in the antrum, then those individuals should receive H. pylori eradication therapy, and they do not need any follow-up at all. In contrast, if persons have extensive atrophic gastritis or intestinal metaplasia, they should be treated for H. pylori, and then they should undergo follow-up every 3 years. Now, these recommendations, again, are based on using normal endoscopy, white light endoscopy. And I think that there's some exciting data that image-enhanced endoscopy, narrowband imaging, confocal endomicroscopy will help us improve our detection of these lesions, allowing targeted biopsies to allow a more precise assessment. What about patients who have dysplasia? Well, if there's a visible endoscopic lesion, if there's not, then there's a grade of dysplasia that can be determined by the pathologist. If it's low grade, treat for H. pylori, follow up within 12 months. If it's high grade, follow-up immediately, and every 6 months after. If there's a visible endoscopic lesion, then we go over to the arm where H. pylori should be treated and the patient should be staged and undergo resection. One last approach that I want to say is that in areas that have a high prevalence of gastric cancer, there may be an alternative management strategy. In these, if a person has a non-invasive H. pylori test that's positive, then one possibility is to do serum pepsinogen testing. Pepsinogens, if atrophic gastritis is present, the pepsinogen 1 level is very low. Patients should then be treated for H. pylori. Cure should be confirmed. If they have a high pepsinogen 1 level, no follow-up is required. They're very unlikely to have gastric atrophy. If they have a low pepsinogen 1 ratio, that's when endoscopy is performed. Biopsies are taken, and if they have mild atrophic gastritis that's not in the corpus and the antrum, no follow-up. If they have more extensive disease, that's when a surveillance program should be instituted. So I'll go ahead and conclude. I think complex human diseases such as gastric cancer are multifactorial. Their pathogenesis combines the effects of microbial, host, and environmental factors. Test and treat strategies for the indiscriminate elimination of H. pylori are not supported by current data unless there's a defined risk factor that's present. Thank you so much for your attention.